Hello, everyone. My name is AJ Keefe, and in this presentation, I'll be covering this paper titled Nuclear Phosphorylated Dicer Processes Double-Stranded RNA in Response to DNA Damage, published in the Journal of Cell Biology. I want to extend a special thanks to the authors for their contribution to this field of research, and I recommend that my viewers pause the video at each new slide and read through the material and analyze the graphics before playing my commentary. If you have any questions, please comment below, and I hope you enjoy. Before jumping into this paper, let's first review this very fascinating Dicer protein. So this protein is simply an endonuclease that cleaves double-stranded RNA into siRNA, and it cleaves RNA into siRNA so it can be loaded into the RNA-induced silencing complex called RISC, R-I-S-C. So Dicer essentially processes double-stranded RNA from something like a virus or endogenously produced short hairpin RNA, and it cleaves it so it can be loaded into RISC for gene silencing. And so that's the canonical activity of Dicer, but it turns out that Dicer is much more interesting than simply getting RNA ready for silencing. For example, Dicer has numerous non-canonical activities, such as directing gene methylation in yeast and RNA polymerase modulation. It also is known to target heterochromatin formation. And more recently, and the focus of this paper, is that Dicer has been recognized in the process of DNA damage repair. So a recent paper by Swahari et al., 2016, found that Dicer knockout in the brain caused widespread apoptosis of developing cerebellar neurons due to the accumulation of DNA damage. In fact, simply knocking down Dicer in cells has been shown to upregulate DNA damage and DNA damage signaling through P53 and ATM. A study by Schlackow and Rickley found that UV radiation would cause the localization of Dicer to DNA breaks, and it mediated the decondensation of chromatin during nucleotide excision repair. So essentially, Dicer was decondensating the chromatin so the repair enzymes could get in there. So Dicer is known as an RNA processing protein, but it's also apparently intricately tied to the repair of DNA. So since Dicer is able to bind and cleave RNA and is needed for DNA repair, it's speculated that Dicer plays a role in the processing of a type of RNA called DNA damage response RNA. So this ddRNA, or DNA damage RNA, is the product of aberrant transcription at sites of DNA damage. So basically, when there's DNA damage, there's also aberrant transcription that produces double-stranded RNA, and it appears to be Dicer dependent. So again, Dicer probably plays some functional role in processing the DNA damage-induced RNA, but exactly how Dicer facilitates DNA repair and what it has to do with this double-stranded RNA that's produced from DNA damage is not very well understood at all. So the primary question tackled by this current paper is, how does Dicer mediate the DNA repair process? And additionally, what post-transcriptional Dicer modifications facilitate that process? So here are the results. The first thing the researchers looked at was Dicer phosphorylation following DNA damage. And they note that Dicer phosphorylation is upregulated across the board following DNA damage. And this phosphorylated Dicer is more prominently localized in the nucleus, where it co-localizes with gamma H2A X. This is a histone variant. It's an H2A histone variant that marks sites of DNA damage. So Dicer was co-localizing with this histone variant that marks DNA damage. The researchers then discovered a specific phosphorylation site at serine 1016 that was necessary and sufficient for nuclear translocation of Dicer. And then there's another two phosphorylation sites at serine 1728 and 1852 that were also discovered to be required for double-strand RNA turnover. So without these phosphorylation sites at 1728 and 1852, the dicer was enzymatically inactive. So then to test how these phosphorylation sites affect dicer's ability to localize and orchestrate DNA damage repair, they transfected fluorescently tagged dicer mutants into dicer knockdown cells. And these dicer knockdown cells by themselves show impaired DNA repair and DNA damage signaling. So transfection of wild type dicer rescues this phenotype, but transfection of dicer isoforms that lack either the nuclear translocation due to the 1016 mutation, or they lacked double-strand RNA turnover by the 1728-1852 mutation. These two mutants, dicer mutants, are not able to rescue the DNA damage phenotype. In other words, dicer is needed for the recruitment of DNA repair enzymes, and phosphorylation at 1016, 1728, and 1852 are all required in order to efficiently orchestrate the DNA repair process. To begin, the researchers assayed the phosphorylation of dicer following DNA damage that was induced by either H2O2 treatment, hydrogen peroxide, or ETO, etoposide. I'm just going to call it ETO from here on. So in this top figure, they fractionated the cells and they extracted the NP, or the nuclear portion, and then they immunoprecipitated all of dicer proteins using this H212 protein or antibody. So they're in this middle lane, they're immunoprecipitating all of the dicer proteins in the cell lysate, and then they separated the proteins out and they stained using a Western blot towards pDCR1. So they're staining phosphorylated dicer. This pDCR1 is a dicer antibody that only recognizes dicer phosphorylations at residues 1728 and 1852. So again, they pulled down all of dicer using H212, and then they stained for phosphorylated dicer, and we see the amount of phosphorylated dicer is upregulated in these cells treated with ETO. So in the ETO DNA damage inducing condition right here, we see increased staining right here. We see this in graphical format over here. So the amount of 1728, 1852 phosphorylated dicer increases by about 2 or 3-fold following DNA damage. And to control for total dicer, they also stained for total dicer right here. And this is A2, it's another antibody towards dicer. We see total dicer is relatively unchanged right here. So this graph actually represents a ratio between total dicer and phosphorylated dicer right here. And so this bottom figure, which looks really intimidating, is actually not as scary as it looks. So what they did was they incubated cells with radioactive phosphate. So any dicer that gets subsequently phosphorylated following DNA damage will become radioactive. So they're going to be any phosphorylated dicer is now going to be radioactive dicer. And so they precipitated all of dicer using this 13D6 antibody. Again, I'm not really sure why they're using all these different antibodies to pull down dicer, but they're using this 13D6 antibody to pull down all of dicer. And it's then they measured the radioactivity of the precipitated dicer proteins in this autoradiographic chamber. So this is just measuring the radioactivity of all the proteins that got pulled down. And so we can see, so ignore these two, this plus condition right here and this plus condition right here for now. This is the CIP condition, it's a phosphatase. Just ignore these two right here. But we can see that phosphorylated dicer is upregulated by about 5 or 10 times in both the ETO positive condition and the hydrogen peroxide condition. So we see Again, if we're looking at these units down here, these are arbitrary units. They're relative to others. So when hydrogen peroxide is gone, there's no DNA damage, and we see that the amount of phosphorylated or radioactive dicer is very low. It's around 2.4 units. When hydrogen peroxide is added, we now have 14.1. So it's about a 10 times increase in the amount of phosphorylated dicer following DNA damage. And we see the same trend in the ETO condition, just slightly less. So there's upregulated dicer phosphorylation following ETO or H2O2 treatment, as indicated by the autoradiographic chamber. We also see a dose-dependent decrease in the amount of phosphorylation following the addition of a phosphatase. So these two CIP conditions are kind of a control, because when the CIP is added, we see decreased radioactivity, which again makes sense because the phosphatase is removing those radioactive phosphates. So this is a control condition to kind of tell us that we're looking at what we think we're looking at, and that is that we're looking at radioactive phosphate groups. Because when the phosphatase is added, we see downregulation of the amount of radioactivity in the dicer that is pulled down. So the summary here is that phosphorylated dicer is accumulating following DNA damage. Next, the researchers wanted to probe what nuclear phosphorylated dicer was actually doing. So in order to more precisely monitor dicer following DNA damage, they took advantage of this highly controllable system called the ASI-SI-ER system, ASI-SI-ER. So ASI-SI is a site-specific endonuclease that reproducibly cleaves DNA at very specific sequences, such as this DS2 or DS1 sequence. This DS1 and DS2 are genetic locations containing the ASI-SI consensus sequence and are known to be cleaved with high fidelity by the activation of the ASI-SI endonuclease. So in order to control this cleavage of these two locations, they fused ASI-SI to the estrogen receptor, the ER, and this keeps ASI-SI sequestered in the cytoplasm. And it's only when 4-OHT is added, such as right here, when 4-OHT is added, it will bind to the ER, to the estrogen receptor, and it induces the translocation of the endonuclease ASI-SI to the nucleus, and it causes site-specific cleavage at places such as DS1 or 2. So the system is used throughout this paper, so let me just reiterate. It's basically, so ASI-SI is a site-specific endonuclease that is fused to the ER receptor, which keeps it inactive in the cytoplasm. And it's not until 4-OHT is added that it binds to the ER receptor, and it causes the nuclear translocation of ASI-SI. And when it translocates, it then cleaves at very specific known sequences. So the primary benefit of using this system is one, you can control when DNA damage happens, and two, you know exactly where it happens because you know the sequence that ASI-SI cleaves at. So in this top figure, we see the result of a chromatin immunoprecipitation of dicer. So they are pulling down dicer, and then they're sequencing the DNA that comes down with it. And it turns out that dicer co-immunoprecipitates with DS1 and DS2 only when they're using these ASI-SI cells that have been treated with OHT. So in other words, these bars right here, where we actually see dicer pulling down DS1 or DS2, these are in the conditions where 4-OHT has been administered and the ASI/SI has translocated to the nucleus and cleaved these locations. So in other words, Dicer is only associating with these two locations when they have been damaged by the ASI/SI endonuclease. So in the bottom figure, we see a dual labeling experiment of phosphorylated dicer. So PDCR1, they're looking at phosphorylated dicer at 1728 and 1852, these phosphoserines on dicer. And they're also looking at the gamma H2AX. So again, the histone variant that marks sites of double-stranded DNA breaks. And they discovered, or they observed, that there's a remarkable degree of co-localization between H2AX and phosphorylated dicer. You can see any yellow in here, this is co-localization. And if you look at just this line right through here, we see this line turned into graphical format. And you can see that wherever there's a peak in red, there's also a peak in green. There's a peak right here. And there's also a peak in the phosphorylated dicer. So this co-localization just suggests that phosphorylated dicer and double-stranded DNA breaks are occurring in the same location. The researchers then screened dicer for specific phosphorylation sites via mass spectrometry, and they identified 7 phosphorylation sites, and one of them appeared to be 3 or 4 times more abundant after DNA damage. So this was serine 1016. It was 4 times more abundant after DNA damage. So they decided to mutate serine 1016 by site-directed mutagenesis, and they transfected these RFP tagged mutant transcripts into wild-type embryonic kidney cells. And so dicer, both wild-type and 1016 mutated, is almost exclusively found in the cytoplasm during control basal conditions. So in this middle lane, we see dicers is almost exclusively in the cytoplasm. And then when you induce DNA damage using the ETO, we see wild type dicer is translocating to the nucleus. Right here, we see abundant translocation into the nucleus, but the 1016 mutant that where the serine has been mutated to alanine, we see that there's no nuclear translocation. And in the top right figure, we see this in graphical format. So the black bars indicate the DNA damage conditions using ETO. And we see that in wild type cells, there's a roughly 3-fold increase in the amount of nuclear dicer fluorescence. So we know it's moving in wild type normal conditions. Following DNA damage, dicer is moving into the nucleus. When serine is mutated to alanine, this S1016A, we see almost no dicer in the nucleus, and it's definitely unresponsive to ETO-induced DNA damage. These other two conditions, S1016D and S1016E, these are known as phosphomimetics that basically mimic a constitutively phosphorylated protein. So these two conditions are showing us what happens when 1016 is constitutively phosphorylated, or at least it looks phosphorylated to other proteins in the cell. And what happens when 1016 looks phosphorylated is that it accumulates in the nucleus, irregardless of if there's DNA damage or not. So we see there's an accumulation of phosphorylated or this dicer in the nucleus, even when there's no DNA damage. So in other words, 1016 phosphorylation alone is sufficient to induce the nuclear translocation of dicer. And the S1016A mutation right here prevents dicer from entering the nucleus following DNA damage. Another phosphorylation site the researchers were interested in is this S1728 1852 dual phosphorylation. And to be honest, I'm not sure why these two phosphorylations are only mentioned together. Perhaps because these two sites are recognized by the pDCR1 antibody that we talked about earlier, as it recognizes specific phosphorylations, but I'm not too sure. But anyways, they mutated these two residues, and as we saw in the previous slide, right here, the S1728 1852 mutation still translocates to the nucleus. So based on from this earlier panel, we know that this mutant still moves into the nucleus just fine. So during their chip sequencing, they noted, this is another earlier finding, they noted that if they degrade RNA using RNase before sequencing the DNA, it prevented Dicer's association with damaged DNA. So this is when I saw this, I thought this was an incredibly important graph. But basically what it's showing us is that when 4-OHT is administered to, induce DNA damage, it causes the association of Dicer with DS1, because this is a chip-seq, so we're pulling down Dicer, and we're sequencing the DS1 locus, which is targeted by the ASI-SI endonuclease. So under normal conditions, when DS1 is cleaved by the endonuclease, because 4-OHT is administered, we see lots of pull down of Dicer. However, if you degrade the RNA after you pull down Dicer in the chromatin, if you then degrade the RNA that's there, we see zero. There's absolutely no pull down between Dicer and the DS1 locus. So in other words, it appears that Dicer requires RNA in order to bind DNA breaks. And so that right there is really fascinating. So the researchers then wanted to find a phosphorylation site that could potentially modify the binding or the processing of RNA on Dicer. And when they mutated the 1728 1852 residues, they observed that double-stranded RNA, indicated by this J2 antibody, accumulated in the cytoplasm right here. So when they mutate this S1728 1852 to A residue, these two residues, we see the accumulation of double-stranded RNA. And then when they supplemented the cells with LMB right here, this condition contains LMB, we see the accumulation not in the cytosol, as we saw up here, but in the nucleus. So when this LMB nuclear export inhibitor is administered, we see the accumulation of double-stranded RNA in the nucleus, telling us that DNA damage causes the accumulation of double-stranded RNA in the nucleus. And so it's probably we are probably looking at DNA damage-induced RNA. And since we know that Dicer binds and degrades double-stranded RNA, we should expect that functional Dicer would extinguish this signal, as it does up here. Because we see RFP Dicer wild type in this condition right through here, we see almost no double-stranded RNA, presumably because this Dicer is catalytically active and degrading all of the RNA. So in other words, this 1728 1852 condition, this mutation, is preventing the degradation of double-stranded RNA. And we see it in graphical format down here. I'll move my cursor up so you can see. We see in the wild type and the S1016A mutation, we see degradation of double-stranded RNA. It's only in the 1728 1852 mutation that we see the accumulation of double-stranded RNA, indicating that this mutation renders dicer catalytically inactive. And the question now is, does this have any functional consequence on the process of DNA repair? And we don't know yet, but we're gonna go over that in the next slide. So in order to assay the effects of the 1016 and the 1728-1852 mutations on the DNA repair process, the authors needed to first better understand the effects of dicer knockdown. So in other words, they needed to know what normal dicer looks like in terms of DNA damage. And then they needed to know what a lack of functional dicer looks like in terms of DNA damage. So they're kind of establishing a baseline in order to compare these mutations to. So for these experiments, they are simply knocking down dicer, and they're seeing what effect that has on the DNA damage signaling and the DNA damage repair process itself. So in this bottom graphic, we see that gamma H2AX, again, the marker of DNA damage of double-stranded breaks, is markedly upregulated in cells that are lacking dicer. So these are knockdown dicer cells. I'm guessing it's knocked down instead of knockout, because I think a dicer knockout will just kill the cell pretty much instantly. So they're knocking down dicer, and they're seeing accumulation of double-stranded DNA breaks. And then in this huge Western blot, we're seeing a pulse of H2O2, hydrogen peroxide. So we're seeing a short pulse. So cells are exposed to DNA damage, and then they're allowed to recover over 24 hours. And then we see that over time, the amount of, in the dicer knockdown cells, the amount of phosphorylated ATM, which is a primary signaling molecule or protein of DNA damage, is upregulated by quite a lot in the dicer knockdown cells. So we see all these numbers, these units are all relative, but we see 0.87, 0.75, 1, and 0.72. We see that in the dicer knockdown. So we see highly upregulated ATM in dicer knockdown. And comparatively to the wild type, we see only 0.61, 0.41, 0.48, 0.49. So we see roughly half the amount of phosphorylated activated ATM when in the wild type condition. And then we also see down here, we see more DNA damage signaling proteins. So phosphorylated CHK1, which is directly downstream of ATM, which it's just another DNA signaling protein. This is an activating phosphorylation. And again, we see upregulation of all of these, or of both these signaling proteins in the dicer knockdown condition as well. So, I mean, these results collectively suggest simply that DNA damage signaling is upregulated when dicer is knocked down. Okay, so I'm excited to finally get into the crux of the matter here. And so in these panels, we are seeing the dual labeling of dicer in red. So any red you see is going to be dicer. And in green, we're going to be seeing either MDC1 or 53BP1. And these are basically the first responders to DNA damage breaks. And so they represent, in these images, the initiation of DNA repair. So wherever you see a green punctate staining, that means there's DNA repair happening at that location. And so remember that these S1016A mutations of dicer prevented the nuclear translocation of dicer, and we can see that here as well in these panels. We don't see we don't see co-localization because the dicer is stuck in the cytoplasm, so obviously it can't assist in DNA repair. So the GF or the RFP tag dicer is not entering the nucleus, And as a result of that, we don't see hardly any MDC or 53 BP punctate staining in the nucleus. So in this condition, the only difference between this graph or this panel right here and this panel up here is that Dicer is not in the nucleus. And we see that when Dicer is not in the nucleus, there's only 8% of cells show punctate staining of 53 BP. And over here we see a similar thing. I'll move my cursor up here. So in the control wild type conditions, the top panels, following OHT4 induced DNA damage, we see 77% of cells with MDC1 punctate staining. So up here, 77% of cells with MDC punctate staining, and we see 58% of cells with 53 BP positive locations. And then when Dicer is excluded from the nucleus with this 1016 mutation, we only see 9% of cells with MDC1 positive foci and 8% of cells with the 53 BP positive foci. So the process of DNA repair is significantly impaired when Dicer is excluded from the nucleus. Okay, so what about this other phosphorylation site, this 1728-1852, that was supposedly required for Dicer's ability to process and degrade double-stranded RNA? Because remember, when this mutation prevented the degradation of double-stranded RNA following DNA damage, and it allowed the accumulation of double-stranded RNA. And also remember that this mutant translocates just fine to the nucleus, and so it fails to degrade double-stranded RNA. So the question addressed in this slide is, does that defect, inability to degrade RNA, interfere with DNA damage repair? And it turns out the answer is yes, or at least it appears. It shows that these panels show us that the inability to degrade double-stranded RNA prevents Dicer from recruiting DNA repair proteins. So although Dicer is now present in the nucleus, it's still not facilitating the formation of MDC1 or 53 BP foci. Again, only 5 and 8% of cells are expressing green punctate staining in the down here in the Dicer mutants. This is the 4OHT condition. I forgot to mention that before. But in the middle panel, these are the cells that have damaged DNA. And we see 77% of cells in the wild type condition, and only 5% of cells in the Dicer mutant condition show green punctate staining. So although Dicer is now finally in the nucleus, its ability to process double-stranded RNA is still required for its ability to orchestrate and mount its DNA repair program. So just to summarize, we learned that Dicer knockdown causes the accumulation of DNA damage, and in order for Dicer to orchestrate that process of DNA repair, it needs to translocate to the nucleus by phosphorylation at residue 1016, and it also needs to be catalytically activated by phosphorylation at 1728 and 1852. So one major question that I'm still left with is how does double-stranded RNA get produced following DNA damage, and how does it recruit Dicer? So in general, I mean, I'm just personally interested in this DNA damage-induced RNA and what purpose it actually serves in the DNA repair pathway. I mean, it supposedly directs Dicer somehow, but I'm not totally convinced by that personally. And I'm still curious as to specifically how Dicer uses DNA damage-induced RNA to recruit DNA repair proteins. And another question I have is, do these mutations that they characterize in this paper affect gene expression? Because remember, Dicer is normally involved in the biogenesis of microRNA to modulate gene expression. So do these mutations deregulate that pathway? And if these mutations don't mess up gene expression, then perhaps you could you could tease out the role of Dicer in DNA repair versus Dicer's role in gene expression. For example, perhaps the 1016 Dicer mutation lacks DNA repair, but retains its function as a microRNA biogenesis. So how might an organism fare when it has this half-functional Dicer enzyme? So does it still result in neuronal death if it retains its microRNA processing ability, but has lost its DNA repair ability? How does that affect the survival of neurons? Anyways, thank you for watching, and thanks again to the researchers for this excellent paper. And if you have any questions personally, please comment them in the comment section below, and I'll be sure to respond. And I hope you enjoyed. Thanks for watching.